Does nobody know why we have an immune system? You know why.
To protect our body from antigens and other cells that may have antigens on them that are non-self.
Yeah, to protect ourselves from cells and antigens, that's kind of all right. Do you want to...
So, like, to infection.
To protect us from infection, fantastic. Okay, so we have an immune system to protect us from infection. That's why it's evolved. It may also be able to help protect us against tumour cells, for example. But evolution developed the immune system to fight infectious pathogens. So we're talking about things like bacteria, viruses, fungi, and parasites. So that's why the immune system had evolved. Now, most of us, most of the time, are not riddled with infections. We all obviously do get infections from time to time. Some of you probably have a cold or some other infection. But most of the time, we're not riddled with infections. So we have the cells and the molecules of the immune system, and Anna told you a bit about those in the previous lecture on the immune system. But most of the time, that's what our immune system looks like in terms of activation. Okay? Nothing really happening. We don't have an infection. We have the cells and the molecules of the immune system kind of sitting there in a quiescent, resting state. They're kind of laid back. They're not really doing anything. Okay? But if we get an infection, then structures on the infectious agent, whether it's a bacteria, a virus, fungi or parasite, there will be molecules associated with that infectious agent that we refer to as antigens. And there are various definitions that one can apply to the term antigen, but essentially it's anything that can be recognised by the immune system. That's the kind of the most basic, easy definition. So you get an infection, there will be structures, there will be molecules, there might be proteins, there might be nucleic acids, there might be lipids, there might be sugars, something that the immune system recognises as being associated with an infection. And I think Anna maybe talked about the whole idea of kind of danger and something that is a threat to us, and the immune system usually does not respond to something that is not a threat, but does respond to things that are a threat. Now, that doesn't always kind of work as well as it should, so there'll be quite a lot of you in the room that suffer from allergy, for example. Now, grass pollen, I mean, grass pollen's not going to hurt you. What's grass pollen going to do to you? It's not going to cause you any harm. It's not like, I don't know, HIV or something that may cause you a lot of harm, or even the influenza virus that could potentially kill you. I mean, people do die of influenza. But grass pollen, cornflakes. Cornflakes, are they going to hurt you? But people suffer from food allergies. People suffer from allergy to grass pollen. So sometimes it doesn't quite work as it should do. But most of the time, for most of us, the immune system does function as we would like it to. It protects us against harmful pathogens, but doesn't respond to things that aren't going to cause us harm. And of course that also includes bacteria and other organisms that are beneficial to us. So don't forget that we have in our guts, we have plenty of commensal microorganisms that actually take up physical space, use nutrients that could be available to pathogens. And sometimes when you use antibiotics, you can actually disturb that balance between the commensal microorganisms and pathogenic microorganisms. Pathogenic, something that causes pathology, something that causes disease. So if you do go from this state of not being infected with something, and you go to that state of being infected with something, then you get an immune response. I don't know if the lights will struggle with this room and the lighting in this room. So anyway, you can see that. So in response to something that is going to potentially harm us, you get an immune response. So what's the point of that immune response? Why we've heard that we have an immune system to fight pathogens. What's the point of the immune response? What's it going to do? have a cold, you have a virus, okay? What would you like your immune response to do? Get rid of it, absolutely. So it's going to get rid of it. If the immune response does its job, it's going to recognize antigens on the virus, or the bacterium, or the fungus, or the parasite. It's going to get rid of it. It's going to destroy it, and then get rid of it. And once it's got rid of it, we're back to the resting state. Okay. So when we get an infection, what happens is that cells and molecules of the immune response recognize that there's something harmful, something pathogenic, something that can cause disease. It mounts an immune response, and it gets rid of it. That's what the immune response is all about. So we can say that antigen drives the immune response. It's a bit like having your foot on the accelerator of a car. You push down on the accelerator, the car goes, you take your foot off, the car eventually will roll to a halt. So antigen drives the immune response. So we have antigen, and this can be recognized by the various cells and molecules of the immune system. One of those cells, a type of lymphocyte, is the B lymphocyte or B cell. It means exactly the same thing. Unfortunately, in immunology, there's lots of terms, different terms for the same thing, and lots of confusing jargon and so forth. We just have to live with that, unfortunately, because it's in all the textbooks, and all teachers will use these terminology. B cell, B lymphocyte, exactly the same thing. Two terms for the same thing. So one of the cells that will recognize the antigen is the B lymphocyte. Another really important cell, which again I suspect Anna mentioned, is the dendritic cell. So this will also recognize the antigen. And many other cells will as well. So one of the things that dendritic cells do is that they show the antigen to the other major sort of lymphocyte, the T cell. So we have two main sorts of lymphocytes, T cells and B cells. As you can see on this diagram, the B cell can directly recognize antigen. It has receptors on its cell surface, actually a transmembrane version of the antibody molecule, that directly recognizes antigen. This is antigen, this is antibody on the surface of the B cell. Simple as that, just directly recognizes the antigen. T cells, at least the majority of T cells, recognize antigen in a rather more complicated way, and they need the antigen to be broken up, if it's a protein antigen, to be broken up into short stretches of amino acids that we call peptides, and these are presented by cells like dendritic cells to a receptor on the surface of the T cell that we call the T cell receptor. So recognition of antigen by B lymphocytes is very straightforward, just a direct recognition, whereas antigen recognition by T cells is a little bit more complicated because there needs to be an intermediate cell, such as a dendritic cell, that shows or presents the antigen to the T cell. Now those two types of lymphocytes, the T cells and the B cells, actually work together to generate the immune response. So there is cooperation between the two types of lymphocyte, between the T cell and the B cell. And particularly a subset of T cells, because not all T cells are the same, so we have different types of T cell. A subset of T cells that are called helper T cells, or TH, cooperate with B cells to activate them. And when the B cell becomes activated, it can release antibody molecules. Talk a little bit more about antibody in a few minutes. Now, I've already mentioned that there are different types of T cell, and one of them are helper T cells, or TH. Another type of T cell is the cytotoxic T cell, or TC. Sometimes, already said, they're different terms for the same thing. Sometimes they're called cytotoxic T lymphocytes, CTL. Cytotoxic T lymphocyte, cytotoxic T cell, TC, all the same thing. And what cytotoxic T cells do is that they kill cells that are infected with a virus. Now, that's quite a kind of brutal thing to do. Our cells become infected, and our immune system kills our own cells. Now, generally speaking, we don't want to have our own cells killed. But there's a problem for the immune system in that the antibody molecule up here can do lots and lots and lots of different things. But one thing it can't do is get inside the cytosol of a cell. It can be taken up into vesicles, but it can't actually get into the cytosol of a cell. And viruses, after they've infected a cell, are essentially hidden from antibody. The antibody can't get to the virus. It's hiding inside a cell. So the immune system, over a huge amount of time of evolution, has had to come up with a strategy to deal with pathogens that are hiding inside our cells. And one of the ways that it deals with that is using these cytotoxic T cells that kill our own cells if they become infected. Now, although it's better if we don't start killing our own cells, if that happens very early on in a viral infection, or any other type of infection where there's a pathogen lurking inside our cells, it's kind of okay. We can cope with losing a few of our own cells. And as long as the immune response is effective in getting rid of the infection, it's okay, we can usually regenerate those cells, and it's not a big deal. However, sometimes you get a chronic infection, and you get quite a lot of killing, if you like, excessive killing of our own cells, because the virus isn't got rid of. The cytotoxic T cells and other cells keep killing our own cells. And sometimes you can end up with the immune system actually doing more harm than good. We sometimes use the term immunopathology to describe disease, pathology caused by the immune system itself. But usually the immune system works efficiently, it gets in there quite rapidly, clears the infection before it's spread to too many different cells, and things are okay. Now, one of the other things that, in fact, virtually all cells of the immune response do, that T cells are particularly good at, is releasing soluble molecules that are called cytokines. Now, there are several hundred different cytokines. You certainly don't need to know about the vast majority of them, certainly for this module. But just be aware that there are these molecules, they're chemical communicators, really, of the immune response, called cytokines. And helper T cells are particularly good at producing cytokines. And they release these soluble molecules. Sometimes described as being like the hormones of the immune system. They release these soluble molecules, and they can influence the activity of other cell types. And one of the things that they can do is influence the activity of another cell type that's called a natural killer cell, or NK cells. And in many ways, the natural killer cell has similar functions to the cytotoxic T cells, in that natural killer cells, just like the cytotoxic T cells, are able to kill virally infected cells. And by the way, one thing I meant to say at the start is just interrupt me as often as you want. If you want clarification, just yell, and I'll answer your questions. So please do feel free to interrupt us often. Now, another cell type, again, I'm sure Anna mentioned this, is a cell that is a phagocytic cell. That means it can take off pathogens, engulf them, and subsequently destroy them. And one of these types of phagocytic cell is the macrophage. It literally means big eater, macrophage. It's a rather large type of phagocytic cell, and it can engulf things like bacteria and fungi. You wouldn't be able to engulf a big tapeworm, for example, because that would be far too big. It would be like you trying to eat an elephant. You just wouldn't be able to. Okay? But you could eat a whole Mars bar just about in one go if you're being really gross. So it can, this big eater can take off, quite large pathogens, but not something that is bigger than itself. And cytokines can help activate macrophages. In fact, many cytokines help activate other cells of the immune response. As you can see here, activating the natural killer cell, activating the macrophage. Other cytokines released by the helper T cells that actually involved in helping the B cell to produce antibodies. And as I've already mentioned, the macrophages can carry out phagocytosis. Macrophages, as well as dendritic cells, these two types of cell are actually quite closely related to macrophage and the dendritic cell. They can also show antigen to T cells. Antibodies can interact with macrophages because on the cell surface of macrophages there are molecules that recognize antibody and bind the antibody to the surface of the macrophage. So if you have antibody bound to an antigen, so if this antibody recognizes this antigen, not shown on this slide, but the antibody can recognize the antigen, and then it can cause the microorganism to be more readily recognized by the macrophage, because the antibody acts like a kind of molecular glue, if you like, linking the antigen to the macrophage, causing it to adhere. Now, another group of molecules in the immune system are a set of molecules that are referred to as complement. And there are about 30 different components of what we call the complement system. And the complement system carries out a number of different activities in helping to protect us from infection. One of the things it can do is it can, just like antibody, cause adherence of pathogens to the surface of phagocytic cells, such as macrophages. Another activity of complement is to cause chemotaxis. This is the attraction of cells of the immune response to the site of the infection. And it's a complement is particularly good, or certain components of complement are particularly good at causing chemotaxis of the other main type of phagocytic cell, the neutrophil. So just like macrophages are phagocytic, so are neutrophils. And we can divide the immune response into really two different types of immune response. The innate immune response, which has very broad specificity for the pathogens and does not have a feature that is referred to as immunological memory. So the things on the left-hand side here, generally speaking, are part of the innate response. Whereas the adaptive, or sometimes referred to as acquired, again, two different terms for the same thing, adaptive response, acquired response. The adaptive response, and the cells and molecules on the right-hand side of this slide really relate to the adaptive response. This differs from the innate response in that it is highly antigen-specific, and it demonstrates a property that is called immunological memory. And what immunological memory means is that the cells of the adaptive response remember having seen a particular pathogen in the past and mount a stronger and faster immune response upon the second encounter with the same pathogen. So this is an antigen-specific response. So let's say you're 3 years old, get sore throat, mum puts you to bed, and a few days later you're well and better again. You had a streptococcal sore throat infection. If 3, 4, 5 years later you get an infection with streptococcus again, during that first infection, the immune system will have learnt that there is this particular pathogen that it needs to be able to mount a really good response against. You've seen it in the past, maybe you'll get the same infection again. And you generate what are called memory cells. And the only type of cell of the immune system that can generate memory cells are the lymphocytes, the T cells and the B cells. So T cells can form T memory cells, B cells can form B memory cells. And these lie in wait in the body, and if you get a subsequent infection with the same pathogen, they mount a stronger and faster immune response that is a defining feature of the adaptive response. So just to remind you, the defining features of the adaptive immune response: immunological memory and a very, very high degree of antigen specificity. Whereas the adaptive immune response does not show immunological memory and is not highly antigen specific. It has broad specificity. So the neutrophils, the macrophages, and most natural killer cells are part of the innate response. They're not highly antigen-specific cells. They do not show immunological memory, whereas the T cells and the B cells, two types of lymphocytes, show this property of immunological memory and antigen-specific receptors on their surface, so have a high degree of antigen specificity. And really, the dendritic cell sits in between these two. Doesn't have immunological memory, doesn't have antigen-specific receptors, so it's really part of the innate response, but its function is to link the innate and the adaptive, because it shows antigen to the T cells of the adaptive response. And in the top part of this other line, this horizontal line here, top part, we've got mostly molecules of the immune response, and in the bottom half, mostly cells, obviously cytokines and also. So one of the most important molecules of the immune response is the antibody. So let's take just a couple of minutes to look at antibody. Before I do that, has anybody got any questions about what I've already discussed? Yes, please.
What's the difference between natural killer cells and cytotoxic T cells?
Okay. So the difference is that the cytotoxic T cells are lymphocytes, and they have on their cell surface an antigen-specific receptor. They are a type of T cells, so they have a molecule called the T cell receptor, and they show immunological memory. So they're highly antigen-specific because they have this antigen-specific T cell receptor on their cell surface, and they can generate memory cytotoxic T cells. Okay? So they're very clearly part of the adaptive immune response. Natural killer cells have a variety of receptors on their surface that allow them to recognize pathogens, but they're not antigen-specific molecules. Okay? And most of them do not show immunological memory. Okay? Now that's the current-ish textbook answer. Okay? But one of the things that makes immunology really, really exciting, but also quite tough in some ways, is that things are always changing, things are always evolving. So about 5-6 years ago, some experiments were done which really shocked the immunological world, because it's been shown that some types of natural killer cells can show something that is a little bit like immunological memory. Can't be, it can't be. You know, they are innate cells. They can't have immunological memory. It's only lymphocytes that have immunological memory. So everybody sort of thought, this is really weird, and maybe thought, you know, maybe it's an artifact. Several other labs around the world have reproduced that now. And because it's not quite like the sort of immunological memory of the adaptive response, sometimes the term trained immunity or learned immunity is used. And in fact, it seems that maybe macrophages also have this kind of trained or learned immunity. So things are really, you know, some really strict paradigms that people thought it's like this and it's like this, are breaking down. And in fact, this whole distinction between innate responses and adaptive responses is kind of beginning to break down. So you don't need to know any details about that, certainly as first-year undergraduates, but it may be worth sort of bearing in mind that things that people think, this is exactly how it is, you know, this is definitely the way things are. It's not always the case. You know, in the light of experimental evidence, sometimes one has to change one's view about things. But essentially there is still this distinction in that they certainly don't have T-cell receptors on their surface like the T-cells do. So that in that respect they don't have antigen-specific molecules on their cell surface. And the type of memory they show is probably not quite the same as the lymphocytes have. But it's kind of worth thinking about a little bit because all of you will have been vaccinated against things. And when you're vaccinated, the whole idea of vaccination is to stimulate immunological memory. You give a harmless form of a pathogen, so it doesn't cause disease, but it generates memory cells. And scientists and pharmaceutical companies that are developing new vaccines, maybe they have to start thinking about this kind of trained or learned immunity that these cells and the innate response have, because maybe it'd be useful to try and boost that as well as boosting memory T cells and memory B cells when you vaccinate. So there's a lot of excitement and interest going on, but don't worry about the details. So that's the answer to your question. Anybody else got anything?
How can we get rid of allergies going up? How can we get rid of allergies going up?
How can you get rid of allergies? That's a very, very good question. And at the end of this lecture, I'm going to tell you that in the second year we have a module, half-unit module called Immunology 2001, IMN2001, all half module, and you could learn all about allergies amongst many other things during that. But essentially, getting rid of allergies is not straightforward, but there are ways. There are what are called desensitization therapies, where you inject very, very small amounts of an allergen and slowly increase the dose. And it seems like you can switch off allergic responses doing that. But generally speaking, the best ways of, or the most common way, should we say, of dealing with allergies don't actually switch off the allergy, but are things like, if you're allergic to nuts, try to avoid any foods that have nuts in them, for example. Or you can take drugs such as antihistamines that will block the response. So there's a lot of drugs that are available to treat allergies, but actually getting rid of allergies is slightly more difficult than dealing with them, if you like, treating them. But there are ways that you can switch off immune responses using what is called desensitization therapy, and that's an area of very active research. Please.
What's the relation of the immune system and cancer?
What's the relation of the immune system and cancer? Now that's an extremely good question. And the way that you can start to ask that question, or answer the question rather, is, well, it's clear that the immune system can fight infection. And it's been discussed for many, many years. Well, as a secondary function of the immune system, can it fight cancer? So how would you answer that question? Well, you could look in patients with defective immune systems. And there are a number of diseases that collectively we call immunodeficiencies, where the immune system is deficient. And sometimes that's due to an inherited gene defect. Okay? So you could look in patients that have a defective immune system and ask the question, do they have an increased incidence of cancer? Because if the immune system controls cancer, well, if somebody doesn't have a fully functional immune system, maybe they'd have those types of people, you'd see an increased incidence of cancer. So the answer to that question, if you take somebody that has an immunodeficiency, do they have an increased incidence of cancer? The answer to that is yes and no. And why it's yes and no is they have an increased incidence of a few different types of cancers, but not most cancers. And the types of cancers that they have an increased incidence of, generally speaking, there are some exceptions, but generally speaking, they seem to be cancers that are linked to an infection. So human papillomavirus causing cervical cancer, for example, Epstein-Barr virus, which can cause lymphomas, Helicobacter pylori that's linked to stomach cancer. There are a number of different cancers that are linked to infectious agents. The infectious agent is part of a process that triggers the development of the cancer. So in those cases, it's kind of, you think, well, yeah, okay, you know, if the immune system's there to fight infection, and certain infections can help trigger cancer, then if you have a defective immune system, you might see an increased incidence of those cancers. For most cancers, it seems that the immune system, does not really do an effective job of getting rid of the cancer. Look, 50% of us are going to get cancer at some point in our lives, So the immune system clearly is not doing a spectacularly good job of dealing with cancer, right? But you can then ask an additional question. Well, maybe the immune system isn't that great at dealing with cancer usually, but can we boost it? Or can we use components of the immune system to treat cancer? And the answer to that question is yes, absolutely. So now there are lots of therapies, and particularly over the last few years, there's been spectacular progress in developing therapies where you boost the immune system or use components of the immune system to fight cancer. And that's referred to as immunotherapy, and that's a very, very exciting area actually of tumour treatment. And there's been huge progress made in the last few years. The drawback really is that it's very expensive. So it's a very expensive type of therapy. But it can, in subsets of patients, it's shown really spectacular results. So there's a lot of research and clinical trials going on. Okay, let's move on in the interest of time. So antibodies, they have a kind of Y-shaped structure. So if you were to remove my head and have this Y-shaped structure, that's what an antibody looks like. 2 arms, and actually consists of 4 chains, which are proteins. They have carbohydrates on them, so they're glycoproteins, which is proteins that have sugars attached to them. You can just see the protein chains here. They have 2 identical long chains that we call heavy chains, because they have a high molecular weight, and 2 identical light chains. So the heavy chains are about 50,000 Daltons, and light chains are about 25,000 Daltons. So the antibody molecule is about 150,000 Daltons, or 150 kilodaltons. So it consists of 2 identical heavy chains, 2 identical light chains. The 2 heavy chains are linked together by disulfide bonds, as you can see here. And each light chain is held to the heavy chain by a disulfide bond. You can divide the antibody molecule up into 2 different regions. The variable region, which varies from one antibody to another, and gives this molecule its antigen specificity. So one antibody molecule might be against streptococcus, another against staphylococcus, another against candida, and so forth. The constant region, which is most of the antibody molecule, does not recognize the antigen, but it mediates what we call the effector functions of the antibody, what the antibody is actually going to do. So it's going to bind to antigen, but usually what antibodies do is, after they bind to antigen, they recruit other components of the immune response. And it's the constant region that does that recruitment of other components of the immune response, as we'll see on the next slide. There are 5 classes of antibody: IgG, IgA, IgM, IgD, and IgE. And those 5 classes of antibody have some properties that are unique to that particular class, and some properties that overlap between different classes. So that's the basic structure of an antibody molecule, and this is what they do. So if you look at the center here, we have an antibody molecule, and sometimes they can actually work on their own. So antibody molecules, by the way, are produced from B cells that, when they become activated, develop into a cell type called a plasma cell. So a plasma cell is derived from a B lymphocyte, and it's a form of the B cell that actively secretes lots and lots of antibody. And these plasma cells secrete antibody, and that antibody can do lots of different things, and sometimes it can work completely on its own. So, for example, it may be able to neutralize a bacterial toxin. So many bacteria cause pathology cause disease by secreting toxic molecules that are called toxins. So, diphtheria toxin, tetanus toxin. And the antibody can bind to the toxin and prevent it being toxic to cells. Antibodies can also bind to the surface of viruses and prevent those viruses infecting a cell. In order to infect a cell, a virus needs to bind to a receptor on the surface of the cell. So if you imagine this is a cell, there'll be molecules on the surface of the cell that a virus will recognise. So for example, the virus HIV binds to a molecule called CD4, you find on helper T cells. So this virus needs to bind to a molecule on the surface of the cell before it infects the cell. If you have an antibody bound to the virus, the antibody gets in the way, and the virus can't bind to the cell surface receptor. So those are two examples of antibodies working entirely on their own, neutralising bacterial toxins, neutralising viruses. So we call those neutralising antibodies. And the IgM, IgG, and IgA classes of antibody are good at doing that. One of the cells that you often perhaps don't immediately think of as being part of the immune response is the red blood cell. And of course, red blood cells carry oxygen around the body, and that's perhaps the major function of the red blood cell. But like virtually all cells, red blood cells don't just do one thing, they can do several things. And red blood cells have on their cell surface a molecule called a complement receptor. They have a particular version of the complement receptor called complement receptor 1. What complement receptor 1 recognises is a particular component of the complement system that is called C3b. And when antibody binds to antigen, so you see the antibody here is this white-shaped structure, the antigen is a blue circle here. When antibody binds to antigen, particularly if the antibody is either an IgM class of antibody or an IgG class of antibody, it activates complement. The complement component C3b is formed, and you have what is called an immune complex. An immune complex is simply antibody bound to antigen. But very often, that immune complex will also contain complement, because when the IgM or IgG class of antibody binds to antigen, complement becomes activated. So it's a rather bizarre term. I think it's lots of weird terms in here. Immune complex simply means antibody bound to antigen. But when IgG and IgM bind to antigen, they activate complement. So often the immune complex additionally contains complement components such as C3b. And this receptor on the surface of the red blood cell recognises C3b. And this acts kind of like an express train, picking up the immune complex, racing through the blood circulation, going to places like the liver and the spleen, where there are lots and lots and lots of phagocytic macrophages that will engulf this immune complex, which includes the antigen, which could be a pathogen, for example. So the clearance of immune complexes by red blood cells is something that's mediated through these two classes of antibody, IgM and IgG. I've already alluded to the fact on a previous slide where I was showing the different components of the immune response put together, that phagocytic cells, if there is antibody, the antibody can bind to antigen and act, I think I use the term act like a molecular glue, linking the antigen to the phagocytic cell. And the reason that the antibody can do that is on the surface of phagocytic cells, amongst many, many other molecules, they also have complement receptors, okay? But amongst many other molecules, they have what are called Fc receptors. Now, the back end of the antibody, this part of the antibody, is often referred to as the Fc part of the antibody. And these Fc receptors can bind the Fc part, or the constant region of the heavy chains of antibody. And this particular type of Fc receptor, there are many different types. This is called an Fc gamma receptor. And that tells us, because it's called gamma, and the heavy chain in IgG is a gamma heavy chain, that this receptor will recognize IgG antibodies. So if the antibody is bound to an antigen, this will facilitate the uptake of the coated antigen, the antigen coated with antibody. We refer to this process as opsonization. It's making the antigen or the pathogen more tasty to the phagocytic cell. And I've mentioned that phagocytes also have complement receptors, so complement can also coat antigens and make them more readily engulfed by phagocytic cells. So this could be a mononuclear phagocyte, such as a macrophage, or it could be a neutrophil. Another thing that happens when complement becomes activated is that you generate components of the complement system that form what is produced a membrane attack complex, MAC, M-A-C, membrane attack complex of complement. And this can cause cells to be lysed. So, for example, if they're infected, complement may be able to help lyse infected cells. Going down to the bottom right here, yet another function of antibody is something that is referred to as antibody-dependent, cell-mediated cytotoxicity, ADCC. And ADCC is a function of the immune system that is exactly what it says it is. Antibody-dependent, it requires antibody. Cell-mediated, it's mediated by a cell. Cytotoxicity, cell killing. And what you have in ADCC is an antibody molecule that is linking a cell of the immune response to a pathogen. And this often could be quite a large pathogen, maybe one that's too big to be engulfed, maybe a large parasite, for example. And you have an immune system cell that, when it's participating in ADCC, is referred to as a K cell. Now, there isn't such a cell type as a K cell. We've heard about natural killer cells, okay? But this is just an operational term for any cell of the immune response that can participate in ADCC. And in fact, nearly all cells of the immune response can carry out this function. Because what is required are two things. The cell needs to have on its surface an Fc receptor so that it can recognize the pathogen that is coated with antibody. And most commonly, not always, but most commonly, it will be an IgG antibody, and therefore it will be an Fc gamma receptor. You also need a cell that can produce molecules that can be released and be toxic to the organism that is being coated with the antibody. And in fact, nearly all cells of the immune response have Fc receptors on their cell surface and can release toxic molecules that can kill parasites and other pathogens. So virtually any cell of the immune response can act as a K cell, a killer cell. And in fact, natural killer cells are really good at doing this. But many other cells can do it. Monocytes can do it. Eosinophil types, cell type called the eosinophil is good at doing it. Lots of cells of the immune response can do it. And then finally, the IgE class of antibody has a very special function, and you asked about allergy, and IgE is, in developed countries, we tend to see the downside of IgE because it's the one, it's out of those 5 classes of antibody, it's the class of antibody that mediates allergic responses. But it didn't evolve to cause us grief from the development of allergy. It evolved because by causing mast cells and basophils, 2 types of cell of the immune response, to release inflammatory mediators, it's a very good way of dealing, for example, with parasitic worm infections. But in most of the world now, where parasitic worm infections are not endemic, obviously there are a few areas where parasitic worm infections still are endemic, but in areas of the world where we've managed to get rid of most parasitic worm infections, we tend to see the downside of this IgE-mediated release of inflammatory substances from mast cells and basophils. So these substances are stored in granules. We use the term degranulation of the mast cell. And IgE is good at sensitizing mast cells for degranulation should the pathogen come along. This could be a pathogen, an antigen from a parasitic worm, for example, be a very beneficial response. It could be an antigen from milk, for example, and not be beneficial but an allergic response to milk proteins. So that's what antibodies do. So we've managed in the first 45 minutes of a 50-minute lecture to cover points 1. So in the last 5 minutes, I'll rapidly go through the last bit. So partly this is a reiteration of things that Anna told you, okay? So don't worry too much. So lymphocytes, two main sorts of lymphocytes, T cells, B cells. They are the antigen-specific cells of the immune system. They define the adaptive immune response. So the only cell that is a cell of the adaptive immune response is the lymphocyte. T cells we can divide into 3 types: helper T cells, regulatory T cells, and cytotoxic T cells. Helper T cells help other cells in the immune system, regulatory T cells regulate other cells in the immune system, and cytotoxic cells kill infected cells. The B lymphocytes or B cells produce antibody molecules, as we've already heard. They have antibodies on their cell surface to act as an antigen receptor. If that antibody is specific for the given antigen that you are encountering, those B cells develop into plasma cells that then release a soluble version of that antibody molecule. Lymphocytes are highly antigen-specific and proliferate in response to antigen. As we've already heard, the B cell has on its cell surface a transmembrane version of the antibody molecule. It directly recognises what we call native antigen. That's just antigen in its natural state. It doesn't need to be processed and presented. Nothing needs to happen to it. It's just directly recognised in its native or natural state, as you can see here. And the transmembrane version of the antibody we can refer to as the B cell set, the BCR. T cells, on the other hand, should see little bits of protein antigen in the form of what we call peptides. So these are short stretches of amino acids derived by proteolytic degradation of protein antigens. And they are shown to the T cells using molecules that are called MHC class 1. MHC molecules. And MHC can be divided into 2 types, MHC class 1 and MHC class 2. In the middle here we have MHC class 1, and this shows antigen mainly to cytotoxic T cells. And if this is a virally infected cell, bits of the virus will be shown to the T cell receptor on the cytotoxic T cell, and this cytotoxic T cell will then kill the infected cell. In contrast, most helper T cells recognize peptides presented by the other type of MHC, that is MHC class 2. Now you find MHC class 1 on all nucleated cells. So this cell here, which is a dendritic cell, a professional antigen presenting cell called a dendritic cell, it has MHC class 1, you see this blue one like it does here, but in addition it has MHC class 2. And the so-called professional antigen presenting cells, which are dendritic cells, monocytes, macrophages, and B cells, they in addition have MHC class 2. And they can show protein peptides to help the T cells. And when the T cell recognises antigen being processed and presented to it, or when the B cell directly recognises antigen, you get activation of what we call naive lymphocytes, ones that have not seen the antigen previously, and those particular lymphocytes divide, they proliferate up in number to produce enough cells to get rid of the infection. Now, those responses that we saw on the first set of slides implied that there's communication between cells of the immune response. And this communication effectively takes 2 forms. Molecules on the surface of one cell interacting with molecules on the surface of another cell. And that requires, by definition, cell-cell contact. Okay? If you have molecules on the surface of this cell, molecules on the surface of this cell, and they need to interact, those 2 cells have to come into contact with each other. So one of the 2 main versions of cell interactions in the immune system is cell-cell contact dependent. And an example of that is this recognition of peptide antigen by the T cell receptor, where the peptide is shown to the T cell receptor by MHC molecules. This requires cell-cell contact. However, we've also mentioned these soluble molecules, cytokines, just one example of many different cytokines is a molecule called interleukin 6. This is released by T cells into the surrounding fluid and then can be detected by B cells because they have on their cell surface a receptor for this cytokine. So you have cytokines and you have cytokine receptors. And for each cytokine there is a specific receptor. So for example, interleukin 1 is an interleukin 1 receptor, interleukin 2 an interleukin 2 receptor, interleukin 6 an interleukin 6 receptor. And by these cell-cell contact-dependent mechanisms and by the release of soluble mediators such as cytokines, these interactions occur, this B cell will develop into a plasma cell that will secrete antibodies. And finally, in the last -1 minute, preventing responses getting too boisterous. I've already mentioned this concept of immunopathology. The immune system generally is a force for good, but sometimes we can respond to what should be innocuous antigens with hypersensitivity or allergic reactions. Sometimes the immune system recognises our own body components. We get autoimmunity that can cause autoimmune disease. In a rather artificial situation, of course, the immune response can be involved in in causing graft rejection. You saw this slide, Anna showed you this. This cell here, the regulatory T cell, has evolved to prevent these types of abnormal responses. So there's a lot of research being carried out on regulatory T cells that can dampen down these unwanted types of immune response. So in summary, antigen drives the immune response, adaptive immune response is involved in proliferation of antigen-specific cells, cytokines aid in communication between cells, and regulatory T cells limit harmful immune responses. And if you found these 2 lectures interesting, think about doing Immunology 2001 next year. So sorry I've overrun, no time for questions. You can always email me if you want to ask additional questions. Thank you very much.